as I said, Ludesi is providing a series of webinars, and this is the second webinar named Quality Controlling 2D Gel Data and Why You Should Do It. Last week we were running webinars on performing the image analysis, and we're also planning to run some webinars on results mining. This is the intermediate step, which is assuring that the data that comes from image analysis is correct and accurate. And since we lost a few minutes in the beginning, I'm just going to skip on through and dive straight in. So the webinar about measuring 2D gel data quality, we have divided into two parts. The first part, we aim to answer the question why we need to do it, pointing out some common pitfalls and also addressing the question what can be used as a relevant metric for 2D gel data quality, and why is it important to use it. The second part of the webinar will be focusing on the question how. How can you do it? Answering the question why and coming to the conclusion that we need to do it, then the subsequent question is, of course, how it can be done. And our aim here is to show you exactly how you can do it. I'm going to give you a free software tool that you can use straight away. No registration, no cost, anything. We're just providing this for free for the community, and we're going to take you through step by step how you can use this to quality assess your data. And as a short summary of that, we can say that anyone would be able to do a quality assessment of the 2D gel data in about half an hour's time. And our goal is that you should be able to do that after today's webinar. So let's start with the first part, why we need to do it. And as a start of that part, I'd like to spend 1 minute on the distinction. And for those of you who followed our previous webinar, you recognize this. We are making the distinction, and I think it could be helpful for you too, to have in mind as you go through the 2D gel electrophoresis workflow, to make a distinction between images, data, and results. So images, the way I see it, and the way it can be described is an outcome or the output from the gel running and scanning procedure. So the laboratory process of running the gels, scanning the gels, eventually results in images and digital representations of the gels that you produced. The next stage of this process is the image analysis process. It takes the images as the input, and it detects spots and match pairs and it quantifies the spots. And basically the output is a data file containing those spots, matches, and quantities. And then the third step is to take this data file of match relations, spots, and quantities, applying statistical algorithms, filters on this. And the output of this is the results. And this is where you're asking your biological, the question to address the questions you have for the experiment that you set up. Okay, so with this distinction in mind, I want to point out some of the pitfalls and problems that can occur if you have problems in the image analysis. And if you just go back to the previous slide for a second, if you look at this type of distinction, you can realize that if you have problems in the image analysis process, causing errors here, you will have errors in the data file. And then you will have errors in the data file where you apply your data analysis algorithms, and this will translate to suboptimal results in various ways. My point now is trying to exemplify some of the ways that this can occur and the consequences it will have for the results you get. So here is one example of a gel set with 9 samples divided into 3 groups. So the left-hand column is group A, and then you have group B, and the right column is group C. And I guess that most people who have experience with 2D gels immediately recognize what the problem is. This red spot outline is swallowing multiple spots at once. So this phenomenon is called amalgamation, where the spot borders encompassing several spots. And the quantitation takes place within the spot outline, which means that this will be detected as one spot with a giant volume. And what happens then in the data file is, if you look at the bar plot here, which is representing the intensity or the volume, the quantity of the spot in each sample, it looks like there's hardly any difference between the groups for this spot. But a more accurate image analysis that actually detects individual spots shows that there's a very clear difference between Group A and Group B here. And so this example to the left, we call with image analysis terminology a false negative. And I want to point out that this is false negative in image analysis terminology. There's not any biological meaning in that. So in terminology that we use to image analysis, we mean that a false negative is a spot that doesn't show up as having a statistically significant expression difference because of errors it has in either spot detection or matching. And conversely, a false positive is a spot that does show up as having a statistically significant expression difference, also because of errors in image analysis. So what's important to point out here is that false positive you will experience this as noise in your data. It will be something that looks like a result, but when you look closer on that spot, you will see that it's a mismatch or it's a faulty spot detection or some other type of error that makes you, having to discard that particular result. The false negative is almost worse because this is something that you won't find in your results. Errors in image analysis will make this spot disappear in the spots that are not statistically significant expression difference. So with that terminology, this image analysis will result in a false negative, a spot that is not seemingly doesn't have a difference between the groups, but with a more accurate image analysis it would have that. Another example looks like this. So here we have two groups. Group 1 is colored in green and Group 2 is colored in red. And the spot that we're looking at is marked with this red spot outline. And again, you can probably immediately see what the problem is. The problem is clear that the spot hasn't been detected at all in Group 1, even though you can see it's there. So for the data file, this means that it would look something like this. It will have very clear intensities in Group 2, but it wouldn't be seemingly not present in Group 1. So this would then show up amongst your results as a potentially interesting spot. But when you look in the image, you can see that, actually it's an image analysis error. It should have been detected. And when it is detected, like you see here, the example to the right, then it shows that the group average of the intensities is almost identical. So this shouldn't be a result. So again, with the terminology that I just defined, this would be a false positive. Something that shows up to be a result, but when you look closer, it isn't. The third example is also based on two groups, 4 samples in each. And the spot we're looking at is marked in the red outline. Here the spot is detected in both groups. But when you look closer, you can see that it's not the right spot that's been matched. So this red spot outline here corresponds to the spot right here. So this is a mismatch. And for the data file, this means that you're comparing the intensity of this spot with the intensity of this spot, which is not the equivalent one. And here it looks like there is a clear difference between Group A and Group B. So when you run a student's t-test or something like that and a NOVA on this, it will look like there is a very interesting difference. But when you look in the images, you see that this is based on an image analysis error. And a more accurate image analysis, meaning correct matching, shows that, again, there is hardly any difference at all between the groups. So this would be, with our terminology, false positive. Okay, so that is some common examples. There are many, many more types of examples like this. And the key thing is that all these types of errors that can occur in image analysis with faulty spot detection and mismatching or faulty quantitation and stuff like that will result in more false positives, meaning more noise in the results. Spots that seem to be results, but when you look closer, it's just based on errors. Or more false negatives, and that is even worse because those are spots that you will never even see, because the expression difference that is actually there is hidden because of errors that have been made. For example, amalgamation is a great example of this. It usually hides a lot of potentially interesting changes. And thirdly, it's also, of course, a huge waste of time and resources to wade through these false positives and negatives.